The DNA replication is a process where a double-stranded DNA is duplicated to create an identical two double-stranded DNA. The mitosis can only be proceeded if the DNA replication is completed. Many enzymes are required to catalyze the DNA replication. Each enzyme has a specific function in this complex process. Please take a note that the visualizations in this video doesn't represent the real structure of the enzymes. The DNA gyrase is an enzyme that relieves any form of tensions that produce after the DNA double helix is unwound. The DNA helicase is the enzyme that is responsible for the unwinding of the double helix by disrupting the hydrogen bonds between the antiparallel strands. The SSBs are the proteins that bind to the outside of the two single template strands and prevent the separated strand of DNA from reattaching. Primase is an enzyme that synthesizes RNA primers using the parental DNA as a template. DNA polymerase III creates complementary strands of DNA during replication. It can only add nucleotides to a 3' of a DNA strand. DNA polymerase I removes RNA primers and replaces them with the appropriate DNA nucleotides. The last enzyme is the DNA ligase that joins DNA fragments together by phosphodiester bonds. The replication of a chromosome begins at a particular sites called organs of replication. Proteins that initiate DNA replication recognize this sequence and attach to the DNA, separating the two strands and opening up a replication bubble. Replication of DNA then proceeds in both directions until the entire molecule is copied. Several kinds of proteins and enzymes participate in the unwinding. The unwound sections of parental DNA strands are now available to serve as templates for the synthesis of new complementary DNA strands. However, the enzymes that synthesize DNA cannot initiate the synthesis of a polynucleotide. They can only add DNA nucleotides to the end of an already existing chain that is base paired with the template strand. The initial nucleotide chains that is produced during DNA synthesis is actually a short stretch of RNA, not DNA. This RNA chain is called a primer and is synthesized by the enzyme primase. In the leading strand, after RNA primer is made, DNA polymerase III starts to synthesize the leading strand. The leading strand is elongated continuously in the 5'3" direction as a fork progresses. While in the lagging strand, the DNA polymerase III adds the DNA nucleotides to the primer, forming an Okazaki fragment. After an Okazaki fragment is completed, the DNA polymerase III detaches. The DNA polymerase I replaces the RNA with DNA, adding nucleotides to the 3' of previous fragment. DNA ligase forms a bond between the newest DNA and the DNA of the previous fragment, and the lagging strand is now complete.